student, today we will perform the estimation of hemoglobin by Sahli's method. This is a hemometer box. There are some material required for processing the practical. This is a hemometer tube box. This is a distilled water, and this is N by 10 HCl, and this is a comparator box. This is hemometer tube, and this is a pipette. This pipette is known as hemoglobinometer pipette. This is a dropper we should use, and this is the stirrer. These are the apparatus we use in the practical. Okay? The procedure to perform this practical is, first we should take the comparator box. Then we should take this tube. In this tube there are the markings. This is a yellow marking and this one is a red marking. The yellow one marking is for gram percentage and the red marking is in percentage. So we have while doing this practical we have to take this yellow marking under observation. In this marking point 2 to 22, this pipette has marking from 2 to 22 from yellow side, and red side it is from 20 to 140 marking. Okay? In this practical we should use this marking, this yellow marking. In this yellow marking we have to take dropper. After taking the dropper we take HCl from the dropper. From dropper we take, from the bottle we take the HCl, N by 10 HCl. We fill this N by 10 HCl in a test tube, that is called the hemoglobinometer test tube, in which up to the mark 2. After that we put this tube in a comparator box. After that we prick our finger. After pricking the finger we suck the blood by the capillary action in the tube up to the 20 microliter. In this tube there is a marking which is up to 20 microliters. There are different pipettes in a hematology lab we use. This pipette is for hemoglobinometer experiment, and other tubes are also, pipettes are also present which is used for WBC count and RBC's count. And this is the tube which is used only for measurements of hemoglobin estimation. In this tube there are some markings are present up to 20 μl. We are picking our finger. After that we suck the blood up to 20 μl mark. After that sucking the blood we have to put this pipette in the test tube. We blow the blood into the test tube. After that we mix this blood with the help of stirrer. After that we mix the solution in the stirrer and we wait for 8 to 10 minutes. After 8 to 10 minutes we should add distilled water with the help of dropper. With the help of dropper, one by one adding, we have mixed these and put it into the comparator box. After that we check first reading, which is darker in, has come. We have to compare this reading with the comparator box readings. In the comparator box there are glass rods are present. These glass rods are used for comparing the color of the test tube. After that we check this first reading. We check from the test tube. It is darker in color. Then we note this reading. After that we add another amount of distilled water in the test tube. we have to add distilled water until the color of the comparator box and the test tube is matched. After matching, we have to check the lower meniscus of the tube to find out the hemoglobin values.